It's a great pleasure to talk about the Ayurveda system, and I'm thankful to the Ayurvedic Medical Association of Russia for organizing this meeting. It's a great opportunity to share our views on Ayurveda. Okay, Ayurveda is a highly advanced medical and surgical system which dates back from ancient times. It has been continuously practiced for at least more than 2000 BC. I mean, it's almost 4,000 years. With Charak and Sushruta Samhitas being foundation pillars on which the whole knowledge of Ayurveda is believed to be. And of course, during these last 4,000, it has been a lot of enrichment of the practice and literature on Ayurveda that has happened. So I'll restart again from where I had stopped. See, the Ayurveda, as I said, has been a very old system. And in the old system, as we understand, like what Charak and Sushruta and other wise men in ancient India had stated, that pratyaksha or direct evidence, anuman or inferential evidence, and yukti are better than shabd praman or textual narrations. You can translate now? Yes, but unfortunately in current times what has happened is that the textual narrations have become more important and they are taken as a matter of fact. And therefore, there is a problem, and that's where we need to reintroduce the philosophy of inquiry and debate. And we should understand, as Professor Martin Mitwede had also stated in Milan Ayurveda Congress, that Shastra is not dogmatism, but is a guidance in the search of truth. So you see, we need to understand the old concepts. As we say, science and technology and healthcare system in ancient India were agreeably ahead of the rest of the world. However, those principles and practices cannot and should not be accepted as such just because they are very old. And therefore, to take advantage of the wisdom underlying the science and technology in ancient treatises, ancient books, and so on, we must dispassionately examine them in light of the current understanding of principles and processes of natural sciences. We need to know not only what it does, but also how it does, and so on. We need to ask more and more questions about it. So if we look in present timings, what has happened is that because of the antiquity and absence of documents, we do not know the detailed procedures and specifications that are there in Ayurvedic literature, like what we have for the current practice in modern science and medicine, where we know precisely what we are doing. And therefore, how Charak and Sushruta and other ancient pioneers of Ayurveda developed the various Ayurvedic formulations and practiced thousands of years ago are not known and may never be known. We need to rediscover them. Well, this is where we now need in-depth studies of the ancient texts in synergy with scientific studies. And that involves participation of actual practitioners of Ayurveda with an open mind. That's most important, that they must have an open mind. And linguists with a good understanding of Sanskrit and other languages used in the classical texts. Therefore, I mean, we need involvement of experimental scientists with expertise in fields like plant taxonomy, plant physiology, human physiology, genetics, cell and molecular biology, chemistry, material sciences, modern medical sciences, and so on. So in that process, we need to understand the mechanism of biological actions of the large variety of Ayurvedic formulations, the various Rasayanas, Bhasmas, Asavas, etc. And that will need systematic and in-depth studies on the mechanism of biological actions of Ayurvedic formulations, understanding of the chemical, physical, and material science perspectives underlying the elaborate procedures employed in preparation of various Rasayanas, Bhasmas, etc. Okay, so one of the important points that we need to consider in Ayurvedic is active principle versus holistic complex formulations. See, modern medicine relies on active principle, whereas Ayurveda relies on holistic complex formulation. This is where we need to examine them in great detail. We need to also understand bioconvex of the actions of the various anupana that is added to various formulations, and basis of various dosha prakritis, innate dispositions in terms of modern physiology, immunology, genomics, and epigenomics. We also need to understand biological base of panchakarma, which is one of the very important Ayurvedic procedures, but we need to know physiological and immunological effects of the diverse panchakarma procedures, and what is the role of epigenetics and lifestyle in response to an individual to a given therapy. And then the most important point is it seems the fundamental philosophy of Ayurveda, Pancha Mahabhuta, which is, they say that 5 basic elements like Akash or space, Vayu or air, Agni or fire, Jala or water, Prithvi or earth, what these are. We need to understand these elements in context of modern science. Besides these philosophical issues, we also need to have precise identity of raw materials and preparatory procedures, like what are the various herbs, which parts are described in traditional Ayurvedic literature, which is to be used, because the taxonomy is not very clear. And then there are seasonal and geographic effects on their efficacies. Depending on when they are grown, they will have different effects. And then the processes and quality control, which is a major issue. So actually understanding these variables is essential for efficient quality control, and we all know that Ayurvedic formulations or many other traditional formulations suffer seriously from quality control issues compared to modern medicine. And therefore, because these being holistic, multi-complex things, I believe that biological quality control assets would be more suitable for these complex formulations than physico-chemical only. And therefore now there is a need to re-examine the traditional philosophies and practices. And this is where the major roadblock for a wider acceptance of the concepts and practices of traditional healthcare systems, including Ayurveda, is that most of the ancient beliefs, practices and statements are taken as fact. And with passage of time, and especially when the texts have been transmitted across generations through the oral rather than written route, as they have been in India, that mostly the passage has been through oral communication, there are always possibilities of mutations, or which may be random or desired, as well as local and temporal variations. Therefore, a critical re-examination of philosophies and practices initiated in Charak Samhita and other classical texts supposedly written several thousands of years ago must not be taken as an insult to the wisdom of these great sages. Therefore, we need to correlate the different languages and philosophies of modern medicine, modern science, and traditional medicine to find what is commonalities. And the problem is Ayurvedic practitioners are not trained in or exposed to contemporary principles of biology, especially the enormous progresses in genetics and genomics, cell and molecular biology, physiology, etc. On the other hand, the practitioners of contemporary science and medical principles, primarily trained in the reductionist approach, find it difficult to correlate the principles and philosophies followed in traditional healthcare systems with experimentally verifiable entities. And this generates justified or unjustified skepticism about many concepts, and there is a lack of trust between the two, and therefore the practice of traditional medicine systems are taken in a different spirit when we need synthesis of the two. Well, these considerations led to the genesis of what we now call in India as Ayurvedic biology, and this happened about a decade ago when a remarkably organized and holistic interaction between scientists and practitioners of Ayurveda was catalyzed for the first time by joint efforts of two people, Professor M.S. Veliathan, who was a cardiothoracic surgeon, and Professor R. Chidambaram, who was a nuclear scientist. And they came together and thought that we need to rationally understand the basic science underlying Ayurveda. And that's where I joined Ayurvedic biology system. So, beginning in Drosophila, I started using this for Ayurvedic biology studies, because Drosophila is one of the most important organs for studies in the field of classical molecular genetics, as also in developmental biology. More important, it has also emerged as a very good model to study various human diseases, including cancer, immune disorders, diabetes, neurodegeneration, etc. And to help in these studies, a large variety of transgenic strains carrying a variety of human disease-causing genes are already available in Drosophila. And there's a short life cycle period and pupil period and adults. And so within 10 years, this may study. So to understand the basic science underlying Ayurveda, we use Drosophila because of the incredibly rich and versatile genetic and other resources, including models for a large number of human diseases available with this. So we have started examining, I'll now share some of my lab's results. We started examining biological effects of two Rasayana formulations, Amalaki Rasayana, or AR as I abbreviated, which is a herbal formulation based on fruits of amla or Emblica officinalis or in Indian gooseberry, and Rasindur, a mercury-based organometallic sublimate or bhasma, which is prepared by a process at organism, cell and molecular levels. And we got these preparations from Ayurveda Kottakkal because to ensure quality control. So now I will share some of our results. See, the Rasayanas are utilized for prevention of diseases, maintenance of health and longevity, and also for treatment of disease. For example, Amalaki Rasayana is a prominent herbal formulation in Ayurvedic classics. It is prepared from fruits of Amla or Indian gooseberry, Phyllanthus emblica, with some other ingredients like aloe vera juice, honey, Indian butter or ghee, through a specific and elaborate process which takes a few months to prepare. It's widely used because of claims that it enhances life expectancy, body strength, intellect, fertility, and reduces age-related debilities. The Rasindur is a different kind of rasayana, which is a sublimate of mercury and sulfur in a stable form with some herbal additions. Preparation involves elaborative steps including precise shodhana or detoxification process. And this is important that mercury and arsenic are generally considered toxic, but they turn into effective therapeutic following the elaborate detoxification process, which includes grinding to convert alpha mercury sulfide into nanoparticles. And this is claimed to balance all the three doshas, the three main body constituent types in Ayurveda as they are recognized. It has a soothing effect and believed to protect body from diseases and aging. It's also considered to be aphrodisiac, tonic, rasayana, rejuvenation, wound cleaner, wound healer, and so on and so forth. And therefore we examined this as well. Now, in one of our first paper published in PLOS 1, what we showed was that if we rear wild-type larvae and adults on Amalaki Rasayan or Rasindur supplemented food, they had formulation-specific effects on different biological parameters like development time, lifespan, fecundity, stress tolerance, etc. But what is important is these beneficial effects generally related to the recommended human uses of the two formulations. Yeah. Now, for example, when we compare the effects of the two formulations, Rasayan and Rasindur, we found median lifespan is increased with Amalaki Rasayan, but Rasindur had no effect. Development time, both of them cause faster development and better larval physiology because salivary gland size and the levels were high. Fecundity, there was an interesting effect. Amalaki Rasayan, when young age feeding, it improved fertility, but adult feeding by itself had no effect. On the other hand, with Rasindur, if the young ones or larvae were fed on Rasindur, their fecundity decreased, but it improved with adult feeding. Thermotolerance, starvation tolerance, oxidative stress, all three improved with Amalaki Rasayan, but only thermotolerance was better with Rasindur than Amalaki Rasayan. Starvation, there was no effect. Oxidative stress on the other hand was reduced, they were much more sensitive. And certain RNA processing proteins like hnRNP, etc., they were increased following Amalaki Rasayan as well as Rasindur, and with greater increase with Rasindur. Important point, mercury was not at all toxic. There was no toxic effect that we could see. Okay, a very recent study on the molecular structure of the properties, physical properties of mercury and Rasindur again confirmed that Rasindur cannot be metabolized in the body to become toxic mercury. This was very important to understand. Yeah. Yeah. Now, having seen these results, we thought we should check their effects on neural models like Huntington's and Alzheimer's, which are modeled in nothing they suppress Huntington and Alzheimer's disease, and we saw that Amalaki Rasayan and Rasindur both enhance their level. Therefore, this was one thing that we saw suppression. The proteasomal activity which takes care of the protein degradation pathway was also alleviated by these three. Cell stress was less, inhibition of cell death was less, and therefore the result is that Ayurvedic Amalaki Rasayan and Rasin's neurodegenerate fly models of Huntington and Alzheimer's disease. Yeah, this is. I'll not explain the details, but basically what this slide shows is that we can activate the desired transgene at a given time in fly models. So we use this model to activate the Huntington's or Alzheimer's disease causing genes in developing eyes of Drosophila, and then examine what changes happen during development if they are fed on Amalaki Rasayan or Rasindur or they are without it. Well, I'm not going to explain these results in detail or this technique, but basically this method allows us to activate the desired disease-causing gene in a given tissue. And so we use the eye as a model system to examine if we activate the Huntington-causing gene or Alzheimer-causing gene in eyes, what's the effect, and how does Amalaki Rasayana or Ras Sindoor will modify this. Okay, just to show you, for example, if you can see my cursor on the developing eye where we express the Huntington causing gene, what we call as 127q gene. This, what you see green here is the Huntington's inclusion bodies, which is actually called the disease. If they are, if the larvae are reared on control food, they show enormous amount of Huntington protein, this green one here in both these. But if they are grown on Amalaki Rasayana, as this middle panel shows, or on Ras Sindoor, the Huntington accumulation of 127q protein accumulation is enormously reduced, and therefore these symptoms are almost gone. Were expressing these Huntington causing disease, they were grown on Amalaki Rasayana or Ras Sindoor food. The inclusion bodies in their neural cells were significantly reduced, and therefore pathology was almost absent. Yeah, then we also saw that the organization of the nerve fiber, the red lines here are the optic nerve, which was completely destroyed in when these Huntington causing genes were expressed. This is what we see in the eyes here, but when grown on Amalaki Rasayana or Ras Sindoor, the optic nerve was much better. The neural cells in the eyes, the photoreceptor cells in the eyes were very well developed, normally formed. Then these are more results showing that the Amalaki Rasayana Ras Sindoor restored the photoreceptor organization much better, and so the adult flies had some vision which otherwise they become blind. I won't go into the explicit details, but the summary is that that formulation feeding made their eyes much better in spite of the toxic expression of Huntington gene. And this adult eye also, that see, insects have this compound eye where each eye has about 700 eyes or so. Each of these rhombus is one eye, and the green ones here are the photoreceptor cells. They're a very precise number. They are disrupted when polyQ or Huntington gene is expressed, but following Amalaki Rasayana or Ras Sindoor feeding, they become again their organization is restored substantially, and therefore these flies have some vision as well. Okay, then there was another way that we can see the enhanced levels of Hrp36, one of the hnRNP RNA binding protein, another hnRNP, which are involved in RNA processing and splicing, and histone acetyl transferase, which increases gene activity. Their levels also go up in Amalaki Rasayana Ras Sindoor, and correspondingly the polyQ, the protein shown in green in my images, is reduced, and therefore the damage is much less. Okay, the other thing that happened was the proteasomal activity, which is the proteasomes are the protein quality control system in cells, which remove damaged and or toxic proteins or those marked for degradation. And the polyQ expressing protein, the Huntington protein, is not easily accumulates. But Amalaki Rasayana or Ras Sindoor feeding improved the proteasomal system, and therefore these proteins were degraded, and that also added to the some of these symptoms. The cell death is one of the major regions for damage in Huntington's and so on, that the neurons die. What we found was that Amalaki Rasayana and Ras Sindoor suppress the induced cell death, but not developmental cell death. And like apoptosis is very important for normal development and differentiation. However, such cell death in response to various cell stress also contributes to aging and age-associated debility. Our findings suggest that, as claimed in traditional literature, AR and RS formulations help in better maintenance of cellular homeostasis, and thereby cells do not die easily with even small inserts like Huntington, and therefore that adds to suppression of the diseases as well as healthy aging, which is one of the suggested effects of the traditional Ayurvedic practices. Then what we also have more recently shown is that Amalaki Rasayana promotes stress tolerance much better, and therefore has anti-aging effects in Drosophila, which agrees with their effects in human system as well. And this is some of our results, that the ROS, which is one of the major components of stress tolerance damage, is significantly reduced when the larvae are fed on Amalaki Rasayana. So, for example, we also saw that the Amalaki Rasayana feeding improved clearance of reactive species. We use paraquat, which is experimentally used to generate ROS in cells, and Amalaki Rasayana fed larvae showed much less the ROS levels compared to the one that were reared on normal food, and therefore this reduced ROS result in less damage to lipids, because we found lipid peroxidation also is reduced, and superoxide dismutase, an enzyme which is involved in removal of the ROS, was very high in AR-fed old flies, which also helps in removal of ROS. So, see, this is another thing that we need to know, is that the heat shock proteins, or which are a specific set of proteins induced when a cell is subjected to a stress, and these proteins help in preventing and recovering protein damage. Since AR feeding makes cells resistant to several different stresses, we asked if this Rasayana enhances levels of some or all heat shock proteins. And our results show that AR feeding did not affect the levels of HSP70 and HSPT3 family, which are most important. So this was a little disappointing, but that's what we saw. But one of the smaller heat shock protein, HSP27, which is an important protein involved in aging and stress tolerance, its level were significantly increased by Amalaki Rasayana, both under normal conditions as well as cell stress condition. And therefore we think this has an important role to play in Amalaki Rasayana induced effects in Drosophila. Okay, now having seen these results in Drosophila, I collaborated with some colleagues who work with mammalian system, with rat system, and what we found was that Amalaki Rasayana improved learning and brain energy metabolism in A-beta, the mouse model of Alzheimer's disease, suggesting restoration of cognitive functions following administration of Amalaki Rasayana in mammals as well. So something what we saw in Drosophila was also seen in mouse model of Alzheimer's. I'll not explain the data of the experiment, but this is the main conclusion. Interestingly, Rasayanas also did same thing when it was administered to mouse models in which aluminum chloride was used to induce Alzheimer's condition. Rasayanas restored memory as well as energetical neurotransmission, and these data suggest therapeutic potential of Rasayanas to manage cognitive functions and memory in preclinical Alzheimer's disease. Then in another collaborative study, we looked at another disease, human disease, Lafora, which is an inherited progressive myoclonal epilepsy syndrome with late childhood or adolescence onset. And what was found was that the Lafora model of mouse, when given Amalaki Rasayana, their seizures were significantly reduced, and they showed much better locomotory function after Amalaki Rasayana feeding. The images in this show the presence of one protein called glial fibrillary acidic protein in the hippocampal region of brain, and this has been shown to be associated with Lafora disease, so that when you see in wild type the levels of this protein are much less, but in Laforin mouse, Laforin KO as it is marked here, this protein level is very high in hippocampus, but when the same mice are fed on Amalaki Rasayana, the protein levels go back to nearly wild type, and this is one of the regions where this seems to be suppressing Lafora symptoms as well. So what we see totally that in Drosophila fly model and mammalian models, what we have seen that Amalaki Rasayana and Rasayanas can improve the physiological state when they are provided, and they can prevent several diseases and aging conditions through better homeostasis and contribute to healthy aging. And this the important point is that maybe these can turn out to be very important procedures for controlling the years and clinical on these aspects in humans need to be undertaken. Mm-hmm. So before concluding, I would like to repeat again to Ayurvedic practitioners that they need to begin scientific in collaboration with science. Is kind of failing in many of the lifestyle diseases. These treatment procedures can be very useful, but we must understand why, how, and when, and that's where research is needed. Principles are what I have been doing, and the Science in Ayurveda initiative has helped us to understand this, and I hope these studies are taken up more and more across the world. I would thank you for this opportunity to talk to you all. Well, I'll be happy to take questions if there are any now. My presentation is over, but if there are questions, I'll be happy to answer them. If can video be turned on now, or it's okay?
Mr. Lakhoti, I think there are no questions.
Okay.
At least nobody's typing. And we would like to also thank you very interesting talk and for the work that you are doing for Ayurveda.
Thank you very much for this opportunity. I hope it's useful. Sorry for the video link not working well, but thank you anyway. And for your translation, thanks.